Okay, so welcome back. We're on video 3 for the chapter on the plasma membrane, so the cellular membrane structure and function. We've finished up the basic osmoregulation, osmosis, and basic diffusion, and now we need to talk about a specialized form of diffusion, and that's called facilitated diffusion. Okay, here's the thing that you must remember when it comes to my world. If I say the word diffusion, you think passive transport. I don't care what word I put in front of it, behind it, in association with it. I don't care if it's facilitated. I don't care if it's passive diffusion. I don't care what word I put with it. If I say diffusion, it's passive transport. We're talking about moving from a higher concentration to a lower concentration every single time. If you remember that, you will not get tripped up on the question that trips up everybody every single year. And then when we talk about it post-exam, they're like, oh yeah, you said that. And I'm like, oh yeah, I did. So if I say diffusion, facilitated diffusion is still passive transport. Okay? It's passive transport, but it's aided by proteins. So this is where we're going to use those transport proteins that we talked about, those channel proteins that we talked about, those carrier proteins that we talked about, mostly the channels though. And we're going to use it to move molecules quickly with their diffusion gradients. Okay? These guys, think of these channel proteins as special hallways, special corridors. You have to have the right pass. You have to be the right molecule or the right ion configuration to get across. That's your VIP pass. But if you've got it, you get to zip from one side of the membrane to the other super fast. Okay? Aquaporins are that specialized channel protein to facilitate the diffusion of water. Facilitated means it makes it easier. It's specific to. Okay? So I'm going to facilitate the diffusion of water. It's still the diffusion of water. It's still from higher concentration to lower concentration, but it's specifically for water. Ion channels in the membrane are another great example of this. Ion channels can be gated, meaning that they'll open and close based on a specific signal, but ion channels will open and close in response to a stimulus, and they'll move specific ions from one side to the other. Still facilitated diffusion, still passive, meaning I'm not spending any ATP. Okay, so the upper image here is a channel protein that is not gated. The lower one is a gated or a carrier protein. Okay, some diseases, as we look at this image, some diseases are caused by malfunctions in specific transport systems. For example, cystinuria, kidney disease cystinuria. So cystinuria is an inherited autosomal recessive disease. We'll get to that at the end of the semester, but basically you got one copy from your dad and one copy from your mom of that gene. So it's an autosomal recessive disease, and what it does is it causes, its most common defect is it causes a defect in the transport of amino acids in the kidneys. They don't move the amino acids appropriately. So what ends up happening is we get a really high concentration of the amino acid cysteine in the urine. And then what happens is we form cysteine stones in the kidney and the ureter and the bladder. And we get an aminoaciduria. So these individuals suffer from chronic formation of cysteine stones. They have a chronic condition. They're more prone to urinary tract diseases and infections. Okay? But it's all based on a defect in this carrier system. What should be moving something from one side of the membrane to the other is no longer doing that the way it's supposed to. Okay, that's passive transport. I'm going to totally shift gears. Well, okay, lie. I'm going to halfway shift your gears, and we're going to talk about active transport. We're still talking about transport. We're still talking about moving stuff across that membrane. But instead of moving stuff by diffusion, but instead of moving it from higher concentration to lower concentration, we're going to talk about moving things in the opposite. We're going to talk about moving things from lower concentration to higher concentration. So the words we're going to use with that is we're going to move molecules against their concentration gradient, or we're going to move them up the concentration gradient. Okay, this is like you went on vacation, you bought a whole bunch of stuff, t-shirts for your family, tchotchkes, stuff for your significant other, your bag was full on the way out, and now you're on your way back, and you got to fit all that stuff into your bag. Okay, you're moving stuff from lower concentration, your hotel bed, into your suitcase, something that's already full, higher concentration. Can you do that effortlessly? No, you cannot. That requires stuffing and poking and getting your roommate or your best friend to sit on your suitcase while you zip it shut. Energy is involved. You're moving stuff against a concentration gradient. You're moving up the gradient, and it's an active transport. It requires energy. When we talk about this biologically in cells, we talk about using ATP. We're going to spend ATP to accomplish this. We're going to spend ATP to move solutes against their concentration gradient. Okay? So we do this with specific proteins typically embedded in the membranes. The big one that we'll talk about is ATPase, or the sodium-potassium ATP pump, but we'll look at that in a minute. Let's get some basic definitions back down and out of the way. I know I said them, but you know I like you to see them in writing. So active transport moves substances against their concentration gradient. Active transport requires energy. It's usually in the form of ATP. Okay? Active transport is performed by specific proteins embedded in the membrane. What this does is it allows cells to maintain high concentration gradients. It allows us to put a great big concentration of some ion or some molecule on one side of the membrane and leaving the other side with very little. Now, I can just hear you out there right now. You're going, but why? Why do we want to do that? You kept saying everything wanted to be in equilibrium. You said equilibrium was our happy place. You're right. I did, and I meant it. But when we create a concentration gradient, we build up a whole lot of energy on one side. We build up a whole lot of a molecule on one side. And what that means is we can now use that high concentration to do some sort of work. We can use that concentration to our advantage. I'm going to show you what I mean in a moment. Our example that we're going to hit here is the sodium-potassium pump. This is one form of active transport. Your cells have hundreds to thousands of these pumps in their cell membranes. This is a really, really huge concept when we talk about biology and we talk about active transport in cells. So this is what happens. Remember on your images that your phospholipid bilayer is there. See that beautiful purple embedded integral protein that's embedded into that membrane. Remember the cytoplasm, the inside of the cell is on the bottom. It's that sandy brown color. The extracellular fluid is on the outside. It's that blue color. Okay, so what we've got here is we have a sodium concentration outside the cell that's really, really high. I have more sodium out there concentrationally than I have inside the cell. And I have the exact opposite in potassium. And what this cell wants to do is it wants to move the sodium out of the cell. It can't do this by diffusion. It can't. It's already got a high concentration outside the cell. It needs to spend ATP to move that sodium against its gradient. So in this sodium-potassium ATPase protein, we're going to bind in 3 sodium. Great, but that's not enough to get the job done. In image 2, you see that ATP come over there? See the ATP in the little splash, that yellow splash? Okay, that guy, we're going to hydrolyze ATP. Remember hydrolysis, remember what we're doing. We're going to break off a phosphate off of ATP. We're going to bind that phosphate to this protein. Because now we've bound something to it, we've changed its structure. Remember what happens when we change structure? We're going to change what it does, and it's going to snap from being open towards the inside of the cell, and we're going to push and we're going to snap to release those sodium out into the extracellular fluid. I just added more sodium to a higher concentration area. I just made it extra concentrated. I spent ATP to do it. But here's what's going to happen. In the process, that now that we're open to the outside, 2 of those potassiums that are at a low concentration out there are going to bind, and it's going to snap back and it's going to bring them back into the cell. I just created a really high concentration of sodium outside the cell and a really high concentration of potassium inside the cell. They're equally 1 plus positive charge ions, but I've now forced where they are on opposite sides of the cell. And that's super duper important for things like muscle contraction. In order for us to have muscle contraction and depolarization of a neuron or a cell membrane, I need to flood sodium into the cell. In order to do that, I need to have a really high concentration of sodium outside the cell. So this is just one example of how I'm setting up these cells in this environment to be able to conduct a nerve impulse. All right, review. On the left, I have passive transport. I have diffusion. I have facilitated diffusion. You see how you have just plain old diffusion through the membrane? That's going to be your hydrophobic molecules, right? Your hydrophobics can cross the membranes, your hydrocarbons can cross the membranes. Next to it, I have facilitated diffusion. That's going to be my my channel proteins and my carrier proteins in that order as you look at them. Remember, these guys are going to be for the hydrophilic substances and specific ions. On the separate image to the right that's labeled active transport, that's where I'm going to spend ATP. I'm going to spend ATP to move molecules against their concentration gradient. On the left in passive, I'm moving them with their concentration gradient or down their concentration gradient, high to low. And on the right in active transport, I am moving them up their concentration gradient or against their concentration gradient or from low to high. Okay, co-transport. Co-transport is why, one of the big reasons why we have active transport. So co-transport happens when active transport creates a really high or a really wide concentration gradient, and we're going to use it so that we can carry something else we need into the cell. So I'm going to create that excess sodium on the outside so I can use its passive diffusion into the cell to bring something else with it. Okay? Plants use this a lot. They use a gradient of hydrogen ions that they generate with proton pumps so that they can transport nutrients into the cell. And it looks kind of like this. So I'm going to use an ATP proton pump and I'm going to pump hydrogen ions out of the cell against their gradient. I'm going to create a super high concentration of hydrogen ions outside the cell. I do this because I need that hydrogen ion to bind to a specific type of transport protein. So I need to get sucrose into the cell. But I don't have a simple carrier protein. I don't have a simple channel protein that's going to let sucrose through. It just doesn't exist. I need another way to do it. And the way I can do it is with that protein there labeled the co-transporter. When that co-transporter binds a hydrogen and a sucrose, it will move both of them into the cell. In order to do that, I need to have them both in a higher concentration outside the cell and a lower concentration inside. It's a passive process. It's a straightforward diffusion process. So we're gonna create that high hydrogen ion concentration outside so that I can use it to drive the sucrose inside the cell. All right. Bulk transport is when I move large molecules, okay? So small molecules in water enter or leave through that membrane, through the transport proteins, everything's good. That doesn't work so well for big molecules. Really big molecules like polysaccharides, big proteins, those guys can't cross the membrane through a simple carrier or a channel protein. It just doesn't work. They're not big enough. They don't fit. It's like parking a semi in your garage. It's not going to work. So what ends up happening is we do a process called bulk transport. Now, bulk transport, it requires energy. Okay, all bulk transport requires energy. We have two forms of bulk transport. We have endocytosis and exocytosis. We're going to start with exocytosis. Exo, what's exo? Exit. Exo is out. So exocytosis, cyto was cell. Remember that from lab exercise 1. So exocytosis is going to have these vesicles or these carrier bubbles are going to migrate to the membrane, they're going to fuse with it, and they're going to push something or they're going to allow something to be released from the cell. Exocytosis, exiting the cell, out of the cell. Okay? This is how a lot of secretory cells will export their products out of the cell. Endocytosis is when I take something into the cell. Endo, in, cyto, cell. The process, the process of bringing something into the cell. Okay. Endocytosis is the reversal or the opposite of exocytosis, and we use different proteins to accomplish it, but it's still a form of active transport. Okay, we're going to look at three very specific types. We're going to look at phagocytosis, pinocytosis, and receptor-mediated cytosis. Okay, so in phagocytosis, that's your top image up here. In phagocytosis, a cell is going to engulf a particle. It's going to get pulled in because it's being surrounded by cell membrane. Then it comes together, it pinches, and it pulls that packet of whatever in. Okay, that vacuole is going to hook up with a lysosome. You're going to learn about a lysosome next chapter. But a lysosome is a specialized packet within the cell that contains digestive enzymes. And then the digestive enzymes are going to release from the lysosome, and we're going to digest what's ever inside that vesicle. You saw this. You saw this with the amoeba and the paramecium. Okay. Pinocytosis is, so phagocytosis is cell eating, is how I think of that. Pinocytosis is cell drinking. And so what happens is I form a little, I keep losing my thing, I form a little pouch here in the cell membrane, and anything that's out here in this extracellular fluid, it's not particular, it doesn't care what it is. Whatever it is going to fill up this sac, it's going to pinch off, it's going to be gulped or pulled into the cell in these vesicles, and then it'll hook up with a lysosome and get digested in that way. This is good and bad. It brings in liquid into the cell very quickly, very easily. It does cost energy, but it's a great way to get liquid in. That's the good part. The bad part is you don't know what you're pulling in. There's no rhyme or reason. It's whatever's passing by, whatever fills up that space first, that's what ends up in the cell. The last one is receptor-mediated. This is pretty cool. So at specific points along this plasma membrane, there are little receptors. There are little glycoproteins and glycolipids here, and they're designed to bind a specific something from the extracellular, okay? A specific molecule, okay? Other liquid comes into this space, whatever else is there, but what's happening here is we want this very specific. So in this case, they want the little purple triangles. And their way of ensuring that the little purple triangles make it in is to have a specific receptor for them. Okay? That little purple triangle in this example is called a ligand. L-I-G-A-N-D. Ligand. L-I-G. Let me see if I can write on here. L, don't laugh, I, G. This is harder than you guys think with a mouse. Okay, ligand. So a ligand is any molecule that binds specifically to the receptor site of another molecule. So these little purple guys are going to bind to the receptors for them. Okay? And what we're going to see is this is a ligand, these purple triangles are a ligand. Then what's happened is the same thing that happened up here. This is going to pinch off and we're going to get this little vesicle inside. But here's the kicker. This is why this is cool. See these coated little protein particles or these little proteins along the outside? They're now on the outside of the vesicle inside this cell. And what that means is there's a very specific signature to this vesicle. There are receptors in the lysosomes, and everybody knows what's in this vesicle because with the ligand receptors, we know that the little purples are in there. Up here, we didn't know if the little purples were in or not. Here, we had no clue if the little purples were in there or not, but because we have this specialized coating on the outside, what's in that vesicle is known to the cell. I think that's pretty cool. All right, so my slides are not advancing. Hang on. I need to close that. I need to do this. Still not advancing. Let's try this. Nope, that didn't work. Hang on, guys. I think I broke it. I'm going to pause and see if I can fix it. Hang on. Okay, I got it back, but I reset it. So I'm going to flip through these slides really quick. Sorry about that. Close your eyes if it makes you dizzy. Really sorry. Almost there. Okay, now I'm there. All right, so here's an example for you to play with. I've got the answers on the next one, but I want you to look at the concentrations of things. So you've got a cell, in there. It's got a 0.03 molar concentration of sucrose and a 0.02 molar concentration of glucose. You know what molar means, right? You know that molar means that it's moles per liter. So you can compare these concentrations. The molar, even if the volumes are different, you have molarity under your belt now. The environment has a 0.01 molar concentration of sucrose, 0.01 molar concentration of glucose, and a 0.01 molar concentration of fructose. Let's say that the sucrose, the glucose, and the fructose no, let's do this. Let's say let's handle this realistically. Let's say that sucrose cannot cross the membrane. So sucrose cannot cross the membrane. Okay? Glucose can cross the membrane. Fructose can cross this membrane. Water can cross this membrane. I want you to figure out what's going to happen with relationship to the contents inside the cell. Who's going to move in and out? Are sucrose going to move in and out, glucose, fructose, or the water? So I'm going to say that again. Sucrose cannot cross this membrane. Glucose, fructose, and water can cross this membrane. All right, I'm going to count to 10 and move the slide, but you're going to want to pause so you can work this out. Okay, here are your answers. So what you're going to look at is you're going to look at this. The concentration of sucrose is greater inside than outside. Because sucrose can't move, what's the water going to do? It's going to move in relationship to sucrose. But that might not be the whole story. Glucose, look at glucose. Glucose has a 0.02 inside and a 0.01 outside. Glucose is going to move out of the cell. There is no fructose currently in the cell. It's out here, so the fructose is going to move in. Now, long ago and far away, somebody taught me that if I add up the molar concentrations of things inside and the molar concentrations of things outside, that will tell me ultimately where all of the water will go. Because inside I have a 0.05 and outside I have a 0.03, water has to move into the cell. Excuse me. For every example I've ever done, that's worked. But it doesn't help you answer the individual questions about glucose and fructose. It'll only answer that big question about the water. So this is the end of the chapter on cell membrane structure and function. I'll wrap it up, get it loaded for you guys, and then we'll get chapter 8 loaded. I'm sorry, chapter 6 and then 7 load. Oh no, this was 7. We'll get this one loaded and then we'll get 8 up for you. All right, I'll see you later. Bye.